Thank you for inviting me. Last time I was speaking here, which was 11 years ago, I was still a postdoc at NYU Center for Neuroscience, and I was probably talking about autism and mechanisms underlying autism. Since then, I moved to industry. I joined a small startup called Gilgamesh Pharma, and so today I'll be talking about using psychedelics to treat depression. So I probably don't need to convince you too much that depression is a huge problem. 8% of US adults experience depression every year. 1 in 5 adolescents experience depression annually. And of course, depression is a huge underlying risk of suicides. And typical treatment are antidepressant medication, SSRIs, which we know, have many side effects. They take a while, 4 to 6 weeks, to actually start working, and don't really work for everybody. So that brings us to psychedelics, right? So what are psychedelics? Psychedelics are small molecules that some of them can be found in nature. When ingested, they create profound sensory alterations, visual distortions, experience of synesthesia, euphoria, ego dissolution, in some people, profound personal insight, mystical experiences, meeting entities, very kind of diverse experiences. So, but why would we, as a company, develop those psychedelics if you can find them in nature, right? So here is an example of, well-known molecule, psilocybin, which you can find in multiple types of different mushrooms. And we know that psilocybin is antidepressant because it went through the clinical trials in depression. But the problem with psilocybin is that if you ingest it, the trip lasts about 6 to 8 hours, right? So it can't really be very easily used in psychiatrist office because someone will be with you for 6 to 8 hours. So they can only do a single patient in a day, right? Not very practical, not scalable. So on the other hand, we have very short-acting psychedelics like dimethyltryptamine or DMT, which can be found in different shrubs, plants in South America. And this molecule, when inhaled, gives you about 5 to 10 minute trip. And we don't know if it is efficacious in depression, but we suspect it is not. So that's why we shuffled few atoms around those compounds and created bradykinin. And bradykinin was designed to have about 1 to 2 hour trip, optimizing for efficacy and duration of the trip. So how do you actually develop those compounds? Those compounds go through what we call a drug development funnel. We design tens or low hundreds of these compounds, and then we submit them through a preclinical assays, which is about 100 different assays that tell us something about pharmacology, metabolism, change of behavior in animals, their safety. And based on the best profile, we really select like 1 or 2 and then advance it into clinical trials, where we first test it in healthy volunteers, figure out if it is safe, and then advance it into patients to figure out if it is actually efficacious for depression. And of course, this is a very lengthy process. It lasts anywhere between 10 to 15 years and costs about $1 or $2 billion, right? And so where I personally work is at the interface between these two, between preclinical and clinical, and this is called translational medicine. When we are actually developing biomarkers that tell us something about hallucinogenic potential, how likely is the compound be psychedelic, and some of these biomarkers can then be also used in humans to figure out if it is engaging the target, meaning the receptors in the brain. So I'll give you some examples. Here is a video of mouse experiencing what we call a head twitch response. It is in a loop. This is an extremely short behavior, about 1/10 of a second. And if we give hallucinogenic compounds to those animals, they will start head twitching. And based on how many head twitches they generate per time, we can tell how likely the compound is hallucinogenic. And of course, this is not translatable to humans. Humans don't twitch or head twitch, but we can ask humans if they're high, right? So that's advantage. We can't ask mice. So what I developed is an AI tool that takes video of multiple animals at the same time, analyzes their body posture, and tells you about those head twitches. Another example is electrophysiology. So we are putting up to 1,000 electrodes into a mouse brain and extracting different features of brain waves, of firing of individual neurons, and then we can kind of triangulate it with known compounds where we know they are hallucinogenic or not. And actually non-hallucinogenic compounds that are activating the receptor are exciting as well. You can feel free to ask me in a Q&A. We can then kind of predict, are they hallucinogenic or not? And some of those features are actually translated to humans, where we can record what we call EEG, that's a surface electrical activity on the head, and we can measure objectively what the drug does to a person. Back to bretisilocin. Bretisilocin now went through a phase 2 clinical study. Now it's in multiple different studies. I won't guide you through all these numbers, but I will point you to a single number, the 94%. So after 2 doses of bretisilocin, this led to a 94% remission, meaning 94% of patients who received 2 active doses went home not being depressed, right? Which is, of course, a huge deal. And it made us all super excited. And not only us, it also made a bunch of large biotech excited as well. So we ended up actually selling this asset to AbbVie at the end of last year. So that's the end of the story of bretisilocin for us. Thank you.